We're going to move on now to our student research showcase. And I would next like to introduce Allison Guitor, who will be presenting on capturing antibiotic resistant genes in the gut microbiome, reveals new benefits of probiotic supplementation in preterm infants. Allison completed her BSc in biology at the University of Waterloo, and during her undergraduate co-op program, she worked for various government agencies and as a research assistant in Dr. Lynn Howell's lab at the University of Toronto. After graduation, Allison joined Dr. Jerry Wright's group as a McMaster student and later decided to pursue her PhD studies there as well. Allison's project focuses on developing and implementing sequencing-based technology to enrich our understanding of antibiotic resistance in the gut microbiome after various perturbations, including antibiotic and probiotic use. So welcome, Allison, and we look forward to your presentation.
Great. Thank you very much for that introduction, Liz, and thank you to the MMRC for inviting me to speak today about some work I've been doing as a PhD student. And I'm in the biochemistry department, so I'm a little bit removed from midwifery, but I'm hoping to give you a view into the world of what I think about on a day-to-day basis. And I am in Dr. Jerry Wright's lab, so we specifically look at antibiotics and antibiotic resistance genes, and I'm hoping to give you a bit of an introduction into that. But in particular today, I want to speak to you about preterm infants. So these are infants that are born at less than 37 weeks gestation, and this means that their gut is underdeveloped as well as various other systems in the body. And in particular, I'm interested in the gut microbiome, which is the collection of bacteria and viruses and fungi that exist within the gut. And we know that this microbiome is really beneficial and often detrimental to health depending on how it develops. With preterm infants, because they are born quite early and have an underdeveloped gut, the progression or development of the microbiome is slightly delayed. And there's been interest in understanding different factors that might influence this development. So things such as when the infant is born, how they are born, whether their mothers are exposed to antibiotics during birth, whether they themselves are exposed to antibiotics, how they are fed. And more recently, there's been interest into probiotic use. All these factors have been implicated in shaping the way that the microbiome develops. Preterm infants, because they're born quite early, they often spend a lengthy amount of time in the neonatal intensive care unit, which puts them at risk of various infections. And they're often given antibiotics prophylactically to prevent these infections. And we know that antibiotics have this effect in terms of reducing beneficial bacteria within the gut. In the NICU, there also is often kind of a reservoir for multi-drug resistant clinical pathogens. So these are bacteria that are resistant to antibiotics, and sometimes they might be resistant to all antibiotics that we have available to treat infections. And so you can imagine if these infants might be exposed to particular pathogens and then given an antibiotic, that one might not work necessarily if those pathogens are resistant to that antibiotic. So this is really what I'm interested in understanding, is what pathogens might already exist in the environment that might be at risk of exposure to these preterm infants, and what resistance genes might they have that allows these bacteria to be resistant to antibiotics. And so in order to study this, we often look at the gut microbiome, so those bacteria that exist within the gut. And in order to do this, we often look at the DNA that exists in there. So the genes that encode proteins, that encode functions within bacteria. And we often refer to this as the metagenome, which is just the collection of DNA that you could extract or get from a complex microbiome sample. In my case, I'm interested in antibiotic resistance. And so this is just a small subset of that information, of that genetic information. And so we often refer to this as the resistome, or the collection of antibiotic resistance genes. To date, there's been over 2,000 genes that have been characterized to be implicated with antibiotic resistance. And so there's a large, we refer to it as an arsenal of resistance genes that bacteria might already encode, or they might be able to acquire. And so this is kind of the arms race that we're currently facing, where resistance genes are becoming mobilized on these genetic elements that can then be spread between different bacteria, different strains. And this is really what's leading to the antimicrobial resistance crisis that we're experiencing, where strains are acquiring these genes and becoming more resistant to all the antibiotics we have available. So understanding this complex dynamic is really important to trying to find new antibiotics and ways to treat infections in preterm infants, but maybe preventing this kind of colonization or persistence of resistant pathogens might be another approach to reducing the risk of antimicrobial resistant infections in preterm infants. So that brings me to kind of the point of my research is how do we prevent these antibiotic resistant infections, or how do we prevent antibiotic resistance in general? And one interesting approach might be the use of probiotics, which are beneficial strains that we can introduce as a supplement to preterm infants. And they've been shown to have other beneficial effects, such as helping to promote a healthy gut development and also reducing rates of certain illnesses such as necrotizing enterocolitis. So we wanted to see if probiotics might also reduce the antibiotic resistance genes that we see in the preterm infant gut. So in order to do this, I was approached by Dr. Jennifer Stearns and Dr. Eileen Hutton and Dr. Catherine Morrison, who are the lead investigators from the Baby and Me and Baby and PreMi studies here at McMaster University. And some of you may be familiar with these studies, but essentially they were initiated to study the impact of various early life factors on the development of the gut microbiome. And so mother-infant pairs were recruited by midwives in Hamilton, Brantford, and St. Catharines. And these infants were followed up to 3 years of age. And for our study, at least, stool samples were collected, so we'd be able to access the microbiome, but there were also various measures of health and development that were recorded throughout the study. So it's a really good cohort to answer some interesting questions about factors that influence the development of the gut microbiome. And in particular, there's been a few studies that have come out from these cohorts, including the impact of intrapartum antibiotic prophylaxis on group B streptococcus, or for group B streptococcus in full-term infants. And then more recently, there's been a study looking at the persistence of probiotic strains in preterm infants. So this is kind of where I came in to expand upon this idea that probiotics may be beneficial in various ways. So for my particular study, we looked at 3 different groups of infants from these 2 larger cohorts. So I have preterm infants that did not receive probiotics. So these I often refer to as NS for non-probiotic supplemented, but they did receive a variety of antibiotics during their time in hospital. And then I also have preterm infants that received both antibiotics, but were also supplemented with a probiotic that is referred to as FloraBABY probiotic. And this contains 4 strains of bifidobacterium and 1 strain of lactobacillus. And these I refer to as PS infants for probiotic supplemented. And then finally, I have some full-term infants that we only have 1 sample for. So this is a 10-day, a sample that was taken at 10 days of age. And these infants were born around 40 weeks, and they did not receive antibiotics nor probiotics. For the preterm infants, we have a longitudinal sampling, as I alluded to in the last slide. So we have samples that were taken while they were in hospital, and then up to 5 months of age. Let me see here. So the first thing that we could look at is the gut microbiome, and this is a really big figure, so I'll try to walk us through it. And this is some data that was generated by a PhD student at McMaster, Efra Yusuf. So what I'm showing you here is the diversity of bacteria that we see in the gut microbiome of these infants. And in the first panel here, this is the early time point, and then moving on to visit 4, which is the 5-month age time point. And so these different bars represent different bacteria that we see in the microbiome. And there's a few things I want to point out to you here. The first one being this purple bar, which represents Bifidobacteriaceae, so a family of bacteria that includes those beneficial probiotic strains that infants were exposed to in probiotic supplementation. So in this region here, I have the probiotic supplemented infants, and then NS again being the non-probiotic supplemented infants. And so you can see that up to 5 months of age, these purple bars become really dominant, but in particular, they're more dominant in the infants that receive probiotics, suggesting again, this kind of supplementation is successful in introducing these beneficial bacteria. Two other bars I want to point out are these, this blue bar and then this kind of salmon colored pink bar. And the blue bar represents Clostridiaceae, which are things like Clostridium, potentially Clostridium difficile. And then in purple or salmon colored here are Enterobacteriaceae, which is a family of bacteria that includes Escherichia coli or E. coli and Klebsiella pneumoniae, which are often implicated in infections and sepsis in preterm infants. So these bars are not the ones that you want to see dominating the gut microbiome, in my opinion. And what we also see is that especially this pink salmon colored bar seems to persist up to 5 months of age in the preterm infants that did not receive probiotics. So this is kind of painting a picture that there is some difference in terms of the gut microbiome diversity. So what I want to look at is, of course, just the antibiotic resistance genes. And so there's a few methods that exist to do this. And the big one that researchers often use is metagenomic shotgun sequencing. So this relies on taking all the DNA from your sample and then using an approach to sequence everything. So you want to be able to read everything that exists in that sample. And so what I'm showing you here on the right is kind of a depiction of what that would be, with the black lines representing just different DNA molecules, different genes. And then the colored lines are representing those antibiotic resistance genes. And in a typical experiment like this, antibiotic resistance is very rare. So when we sequence all this information, most of it is just the background in the microbiome. And then resistance genes are usually less than 1% or less than 0.01% of the data. So we have to sequence a lot. It becomes really expensive. And when we're doing these large cohort longitudinal studies, it's really not feasible. So what I do in my PhD is an alternative approach, which is to specifically enrich or capture those DNA molecules that are associated with antibiotic resistance. And this allows us to enrich a pool of DNA, which we then sequence, and it becomes much more manageable in terms of the analysis and the amount of data that you generate. So I can do this method, and I did it on stool samples from our preterm probiotic supplemented infants, or non-probiotic supplemented infants, and then full-term infants. And I can compare the resistance genes that I see in these different groups to try and see if there's any difference. And so what I'm showing you here is just the number of resistance genes that I found in one, or each dot represents an individual infant. And then I separated it based on the 3 cohorts that I have. And so this is at an early age for the preterm infants and at 10 days of age for the full-term infants. And you can see that there's this, in a few of the infants, we see that there's an increased number of resistance genes that we only see in that cohort, suggesting there's some difference in the diversity of resistance genes that we see in infants that did not receive probiotics versus those that did. So this is just a counting, how many genes do we see? But what I think is more relevant is what are those genes and are they important? Should we care about them? And so I'm going to show you a heat map here of what these genes, or which genes we see in one group versus another. And again, this is just at one time point at an early age. And I've broken it down based on different resistance gene families, which may not be familiar to everyone. But for example, these first 4 are genes that confer resistance to aminoglycosides. So if you're familiar with gentamicin, which is often used in preterm infant care, the next ones here are beta-lactam resistance genes. So they would confer resistance to things like penicillins, and then also some of the later generation beta-lactams that we now use in the clinic because resistance has become more prevalent for those. And then at the bottom, these are genes that confer resistance to vancomycin. So really this is painting a picture that infants that did not receive probiotics have this increased diversity of resistance genes in comparison to probiotic supplemented infants as well as full-term infants. And you can see that even the probiotic supplemented infants look a bit more similar to the full-term infants in terms of the antibiotic resistance that we see. So again, this is just one time point, but we also had those longitudinal samples for the preterm infants. And again, I can look at the number of resistance genes and compare it at all the different time points from in hospital up to 5 months of age. And you can see that there does seem to be this increased diversity that persists up to 5 months of age in terms of the number of resistance genes that we see. And again, I can look at what those resistance genes are, what antibiotics they confer resistance to, and look at it up to 5 months of age in the preterm infants. And what I see is that there are certain resistance genes families that we only see in infants that did not receive probiotics, and that these persist up to 5 months of age in the infants that did not receive probiotics. So suggesting that there is some difference in the diversity, and that probiotics at an early age have some effect in terms of reducing that diversity up to 5 months of age. Finally, I just want to pinpoint, if we look at these resistance genes and I try to match them to a particular bacteria that might exist in the gut microbiome, this is what I'm showing you in the next slide, is basically the interactions between a resistance gene and a potential bacterial host. So if I find this resistance gene, can I say which bacteria it might be coming from? And so in circles here are the resistance genes, and then in squares are potential bacterial families that I maybe could identify this resistance gene in association with. And so this is the picture for infants that did not receive probiotics, preterm infants that didn't receive probiotics. And then on the right here, I have the picture for the preterm infants that did receive probiotics. And you can see that there's just much reduced diversity in terms of the resistance genes that we see, but also the bacterial families that we see, really suggesting there is some stark difference between probiotic supplemented infants and non-probiotic supplemented infants. And I just want to zoom in on one interaction here, which is this SHV gene, which is a beta-lactamase. So again, confers resistance to penicillins and first generation cephalosporins, and its interaction with Enterobacteriaceae. And so what I'm showing you on the next slide is perhaps a lot of information, but this is my attempt to track this resistance gene in different infants at different time points. And so in purple here is that particular resistance gene. And in many cases, I can see kind of the genetic context. So that gene and then what's in front of it or behind it. And using that information, I can track to see if that gene is in the same kind of context in that infant's microbiome over time. And what this tells me here is that, first of all, this is unique to the infants that didn't receive probiotics. So I don't see this in the probiotic supplemented preterm infants, but you can see that a lot of these infants had this resistance gene while they were in hospital, and then it seemed to persist up to 5 months of age, suggesting that whatever organism is carrying this resistance gene, it is persisting up to 5 months of age. So the SHV beta-lactamases are intrinsic to Klebsiella pneumoniae. Again, this is that Enterobacteriaceae family that I was talking about. And the risk of this is that these resistance genes might be found on mobile genetic elements and may have the potential to spread to other bacteria. And really what I'm getting at is that this exposure likely happened in hospital, is unique to the NS or the non-probiotic supplemented infants, and that this particular resistance gene and its host are persisting in the preterm infant gut. So in conclusion, I'm hoping that I was able to get some messages across today in relation to the development of the gut microbiome of preterm infants, and that given their early birth and kind of compromised immune system, they're often given antibiotics in the NICU, and this has negative effects on the beneficial bacteria that we see in the gut microbiome. We also know that preterm infants are at risk of exposure to certain clinical pathogens, which could often lead to sepsis or other infections. And so this exposure often likely has a negative impact on the health of the preterm infants. And then, of course, what I've hopefully explained a bit more today about is that antibiotic exposure can select for these bacteria that are resistant to those antibiotics, and then that might provide an opportunity for those bacteria to persist in the preterm infant gut. What I'm hoping I've shared a bit more about today is that probiotics are known to have beneficial effects on the development of the gut microbiome of preterm infants, and that perhaps one of the mechanisms of probiotics might be in reducing the prevalence of certain multi-drug resistant clinical pathogens. And this could be an indirect or direct competition between probiotic strains and potential bacterial pathogen strains. And so I hope that probiotics might be considered as a means to help not only in the development and health of preterm infants, but they might have this added benefit of reducing risk from multi-drug resistant clinical infections. And so that leads me to the end of my presentation. There's many people I'd like to thank, in particular, Dr. Eileen Hutton and Dr. Catherine Morrison for giving me access to these samples from the Baby and Me and Baby and PreMi studies, and in particular to the midwives from Hamilton, Burlington, Brantford, and St. Catharines here in Ontario for recruiting the patients that were involved in these studies, which allows us to kind of answer some really interesting research questions. Dr. Jennifer Stearns and Efra Youssef were very instrumental in helping me understand the preterm gut microbiome. Amos from Dr. Andrew MacArthur's lab was instrumental in doing the analysis and helping me develop an analysis pipeline to do this research. And of course, my committee members and the McMaster sequencing facility for helping me generate all this data. And finally, the funding that was involved in the initial Baby and Me and Baby and PreMi studies, as well as my own funding. And thank you.
Thank you so much, Allison. That was a really wonderful presentation. And I feel like as somebody it's been a long time since I've taken basic science courses, you did a really nice job of making it very understandable. And it's certainly easy to see how this research could have really direct impacts clinically. So it was lovely to hear. Just for our audience, if anybody has questions for Allison, you can add them into the Q&A box. Then there's also, I think Rash has put up the poll as well. So if people are willing to provide feedback, we'd love to get your feedback. It looks like we do have a couple of questions here. So one of the questions I think was around the preterm infants and just how when they were discharged and how long they were, I guess a general sense of how long they were in hospital for.
Yeah, so in terms of how long they were in hospital for, I don't have the numbers off the top of my head, but I know that they were followed. So the data that I showed you today is up to 5 months of age, but we are looking at some clinical data up to 2 years to see if there's any differences in terms of their health. And in particular, I'm interested in, did they have infections later on in life, and were they given antibiotics later on in life? So that's hopefully some information we can get out soon.
Great. Another really interesting question from Cecilia Javet. Was there any analysis about the preterm infant feeding and drug resistance? So did all of the preterm infants receive manufactured formula? Did all of them receive some breast milk? And so she's just wondering about the impact of maternal antibodies in the preterm gut and how that might impact what you're looking at.
Yeah, so I haven't looked at that in particular. I know that many of them were breastfed. So I think that would be something that I could look at, trying to kind of separate the small sample set that I have and see if there's any difference in terms of breastfeeding versus formula fed, but I haven't looked at that in particular.
Great, thank you. I'm just trying to check and see if there are any other questions that I have missed. And lots of other good conversation going on in the chat about our earlier presentations too. I think that's it. We'll just wait another minute and just see whether there are any other questions before we wrap up.
Sure, thank you.
Maybe you could tell us a little bit about if there is any research that you're aware of that's looking at doing any kind of larger studies of this in a clinical setting now.
Yeah, so there is one, I believe, that's looking at probiotic supplementation in terms of reducing rates of certain infections. And so I think in particular, I'm interested in antibiotic resistance. So it kind of goes hand in hand. If you could look at different, I guess, factors and outcomes in that sense. I forget the study off the top of my head, but if I figure it out in the next 5 minutes, I'll put it in the chat for people.
Great. Another question, I'm not entirely sure, maybe this might make a bit more sense to you, Allison. Question about, does this cause encopresis?
I'm not familiar with that term myself. Let's see. I guess impacting stool, passing of stool, perhaps. I don't know off the top of my head.
Okay. We do have a few other questions about the probiotics. So how long were they given to the neonates? And also, was it a particular probiotic brand that was being used? I think BioGaia is something that some people are familiar with using, and so they're interested in whether it was a different one.
Yeah, so the probiotic that was used is called FloraBABY, FLORA-BABY. And I believe it's manufactured here in Ontario, so it's a Canadian brand. And I guess some background on why probiotics were given is that it was kind of a clinical change in the McMaster NICU, where infants that are born at less than 34 weeks gestation and less than a certain birth weight, I think it's 2 kilograms off the top of my head, they were given probiotic supplements. So from as early as they could, I assume, and they were given probiotics every day, so a supplement every day. And I guess they were given until discharge, and then some parents or guardians had the option to continue supplementation after discharge from hospital. And so there is kind of a variability in terms of the length of probiotic exposure for these infants.
Okay. And just another question, I don't know whether you answered this previously, but do we know how long these babies were in hospital?
I don't have the numbers, unfortunately.
In front of you?
Yeah. But I think it is a little variable in terms of their discharge or when they were discharged. Yeah.
Okay. And I guess the question there was wondering whether or not you had taken any look at whether there were differences in the gut flora depending on the length of stay.
Yeah, I guess that's maybe getting, one of the things that I'm interested in also is, were they in the hospital at the same time? Were they exposed to the same flora in the hospital at the same time? And does that kind of impact what we see in the gut microbiome? And I don't have the exact dates for whether they overlapped in the NICU at the same time, and maybe you would see some strains that are similar. There are a few twins in the study too, so that's another interesting avenue to look at, is whether the twins have the same flora that exists, but obviously it's a very small sample size. So I'm looking at very individual cases of microbiome development.
Really interesting though, and lots of great findings to develop new hypotheses that you can go on to test in with bigger data sets, right?
Yes, for sure.
Super. Okay, thank you so much, Allison.